Hi everyone. I'm ready to discuss with you this scientific article for coming exam major in Edexcel Unit 5 exams, right? So we have two articles this time, Article 1 and Article 2. So today is 6th of May, so I'm publishing this before this exam. So I try to make a full discussion about from this video. And also, you will have separate PDF file with the questions and the model answers or answers marking scheme points. So I need to tell you something before make these exact questions. This is all about the Y chromosome. Now think about what is a relevant Y chromosome to Unit 5. Where do you learn about the chromosomes? Where do you learn about the cell division? Where do you learn about the mutations? Where do you learn about uh, the the meiosis and mitosis and all these things? Mostly, you have learned the basic biology in AS, so it's better. You have a time now, a little bit revise the main component of the AS part, which is relevant to the chromosomes and genetics. And here, the main part of this biology lesson is the heart failure. I mean, what we learn about the heart is the direct theory. Rest is all about the Y chromosome. So we all know homologous chromosomes. They are pair of chromosomes, and this pair of chromosome usually not identical. So they are not they're same shape, same height, and same position of the centromere. The many things are common to them, but there is no reason to have the same gene, same allele on the other side. But when it comes to the karyotypes, you will see karyotype is all chromosomes on human. The diagram, you can see X and Y. So X and Y is not homologous chromosomes. So X and Y is not homologous as their length is different, as their centromere position is different, as they have many different genes and not the same. So they are not homologous chromosomes. And the Unit 5 main lessons are respiration, internal environment, coordination, and gene technology. So when we making the questions, so internal environment, homeostasis, learning about the heart rate regulations, conductions, all these things are the main component, should be the main component for the questions. Because if you look at the all past questions from scientific article, most of them are coming from Unit 5, not Unit 4, not AS level. So then we have to stay with most of our questions in this background, right? So I want also to give some information about lessons which you haven't learned. You heard the word, but not learned. Chromosome inversion. So you will see in this article, chromosome inversion is one of the main topic here and there. If you look at, they discuss chromosome inversion, gene loss, chromosome inversion, gene loss. So chromosome inversion is the phenomenon we try to learn when the part of the chromosome is break and then it turn 180 degree, that means invert, and then rejoin with the chromosomes. So chromosome, part of the chromosome break, 180 degree rotate, and then join with the chromosome. So here you will read the note, what is chromosome inversion? So, and uh, do all the genes in Y has on X? For example, when you take homologous chromosomes, the gene on one chromosome, also find in the other chromosome, but different versions. But that gene, same gene, for example, the eye color gene. So eye color gene is this chromosome and also this chromosome. These are homologous chromosomes now. But there could be different versions. Now, important thing about this, the X chromosome and Y chromosome, you can see here. So look at here, the only some of the genes which are common to X and both Y. X and Y common. The rest is different to X and Y. So even Y chromosome here, differential region of the Y, I mean different genes, and here different genes. So only some of the genes are common to X and Y, and most of them are not common. Do both X chromosome express genes? Because X chromosome, when two X chromosome come into female cell, do both express? No. One is inactivated. So it's called X chromosome inactivation. Surprisingly, you have learned this. You have learned this, but I think you're completely lost. Where did you learn? So AS, so I showed here, chronic villus sampling, the genetic testing, CVS, disadvantage, two disadvantage of CVS, 0.5 to 1% spontaneous abortion, and second one is mentioned in textbook, all parental paternal X chromosome are inactivated. So just sensed by any problem of genes that they can't be checked by, we cannot genetic screening. When they're inactivated, we can't use them. So these X chromosomes are inactivated is mentioned. That's also very important thing, X chromosome inactivation. So coming back to this whole article, in summary, 200 to 300 million years ago, when phylogenetic trees, all the organisms are evolving. So, you know, basically the reptiles are the most primitive type of organisms. Reptile, then the birds, and then the mammals came later. So 300 million years ago, there's no gender. There's no gender. The gender evolution started somewhere around 200 million years ago. And before that, there are environmental factors. They changed the gender. But what happened? One chromosome started to do more mutations, more changes, and try to deviate from its X chromosome. So when you talk about the X2 X chromosome, maybe this 2 1 X chromosome try to change very fastly, and it doesn't match with the X chromosome, it becomes a Y chromosome. So this whole evolution of XY chromosome is the basic for this research article. And in the future, the dynamic changes of the Y chromosomes take place still. It's going on right now and it will change in the future. And some predict, okay, 5 million years ahead, 5 million years later, maybe no Y chromosomes because it's changing too much. And these changes are too fast that will disappear. And some says, no, the changes are actually evolving and new genes will come, it will stay. So paragraph 1, the immune cells of many older men lose their Y chromosome. First question we are here. Why is Y chromosome loss occurring mostly in immune cells? So answer is Y chromosome deletion occurs more frequently in immune cell as the Y chromosome does not participate in any function in immune cells. Number one, Y chromosome has no distinct function in immune cell, and immune cells has high rate of division. These are the 2 points. Next, paragraph 2. Although some people have other combinations such as XXY or XYY, so these are the two common conditions, two X chromosome and Y. That's called Klinefelter syndrome. And 1X and 2Y is called Jacob syndrome. There are people among us with these two XXY and XYY. Although some people have other combination, XX and XXY, now why people have this kind of a combination? So the question is, explain why certain individuals possess different combinations such as XXY or XYY. Now, it's easy to understand when the gamete form, right? When the gamete form, meiosis 2, meiosis 1, meiosis 2, and meiosis 1, metabase 1. The X chromosome, let's say this is two X chromosome arranged at the equator. So then one is going to the other side, other one is going to the next other side. So then they separate. If this two X chromosome cannot separate and they move into the one cell, that gamete has ovum has 2X. And in male, let's say, so again, two Y chromosomes. After duplication of this two Y chromosome, they should separate. But this two Y chromosome somehow not separate and they go into one cell. That means one gamete form with two X cell, one gamete form with two Y cell, Y chromosome. So then if they fuse, it carries extra chromosome. That's a 47 chromosome. Because it is unable to separate from the meiosis. So here, during meiosis, homologous chromosome fail to separate properly, resulting in one cell receiving an extra chromosome while the other cell lack it. So when one cell get two, other cell doesn't get one. So this occurs when X cell with two X chromosome is fertilized by a sperm cell with a Y chromosome, or when a sperm cell with both X and Y chromosome fertilize the normal X egg. So sperm has X and Y and fuse with X cell with XXY. So this is the answer for the question. Stem cells that produce immune cells. Third paragraph. So this is a common question. Explain how stem cell develop into immune cells. So I think explanation is stem cell get a stimulation, chemical stimulation, which send a signal to the nucleus. So then epigenetic modifications, then switch on and switch off certain genes, switch on gene transcribe and translate to produce proteins. Protein permanently modify the cell. So this question repeatedly asked in many times. Marking scheme result I am showing you October 2023. Check October 2023 scientific article, unit 5 scientific article. Similar question has been asked. So there is again chance of asking this question. So I included these points again because this prediction of 2023 same paper I clearly predicted this question can be asked, the same question asked. There's a chance again asking the same thing, and marking scheme points are given, mostly AS knowledge. But I again explain, I explained this one, and here you can see the marking scheme point. Fourth paragraph. I didn't highlight anything yet. Then fifth paragraph. Use the CRISP gene editing technique to remove the Y chromosome. CRISP gene editing technique is not in our syllabus, that is in the gene technology, but Cambridge syllabus, they have CRISP gene editing technology. I don't think that something will come up in a CRISP because it's theories. You haven't learned the theory. But for the safe side, I will explain you what is CRISP. The CRISP is a recent discovery of the new gene technology that came after recombinant technology, gene technology. And now we have a new technology, CRISP, cluster regulator interspace short palindrome repeats is the word for that. So what we do, we make a small complementary RNA sequence. Let's say we have a target gene. We know the sequence. Then to the complement to that sequence, we make a small RNA complementary sequence. Then after that, that's called guide RNA molecule that is designed to match specific DNA sequence in the target gene. So that's a target in the DNA. Second, we are coupled to this small guide RNA an enzyme called Cas9. So Cas9 is incorporated into this guide RNA, and then a guide RNA specifically bind to the DNA. Cas9 cut this part of the DNA. After cutting, we can remove the mutated one, we can insert correct one, or we can remove the mutated one, or we can add new sequence. So that's called editing. So in recombinant DNA technology, we just deliver gene that express the protein. But here different is, so probably there's a question will come. The CRISP technology is gene editing technology in which they can do this. Compare and contrast recombinant gene technology with CRISP. So there's a high chance that you will get these questions. You will be asked to compare and contrast, and you will give what is CRISP. The CRISP is like this it works like this, and compare and contrast with what you learn the traditional gene technology, ligase. We cut with restriction enzyme, then add ligase, vectors, and then we deliver the gene. But here is different. We have a guide RNA, we have a Cas9, and we have a specific target. And after we remove that one, we can do many things. We can insert one, we can delete one, we can make a new one like this. So that's the editing of the gene. The third part is editing of the gene. So carefully read these three to understand better. If the comparison question comes, you can better compare. Okay, here they use guide RNA, they use Cas9, and they can do editing, and they can do removing and inserting many genes like this. Paragraph 6. Scarring of heart muscle can contribute to some common type of heart disease, such as heart failure, says Forsberg. So scarring refers to the replacement of heart muscles with the connective tissues. So that means if this happens, then most of the heart muscles are replaced with the non-functioning tissues. Explain how scarring contributes to heart failure. Number one, scars reduce heart muscles. That means the scarring remove the heart muscles, cardiac muscles, replace with the connective tissues. Then the left ventricle lost a lot of muscles and force of contraction is less. So they cannot pump blood, so cardiac output is less. Then there's another question can be asked in the same place. Explain how scarring of heart muscle detected. How can we detect whether heart muscles start to malfunctioning? That means they are not properly functioning. So then you can propose, you can do ECG, because scar tissues which replaces damaged heart muscle can disrupt the heart electrical signal, leading to changing ECG waveform. So similar question can become ECG and detection of this kind of thing, or early diagnosis of this. Next, paragraph 7, triggering the release of an inflammatory signaling molecule called transforming growth factor beta. So inflammation release many cytokines, including transforming growth factor beta or TGF beta. Explain how transforming growth factor beta causes scaling. So you will write your answer. Immune cells lacking Y chromosome can contribute to heart damage by promoting scar tissue building up in heart muscles. Specifically, macrophages, a type of white blood cell without Y chromosome, can stimulate cells in the heart to produce more connective tissues, resulting in increased scaling. So, one hand, we are talking about Y chromosome is not necessary, but when the Y chromosome lost, the imbalance will come. It resulted in enhanced inflammations. So we don't know how Y chromosomes actually involved in inflammations, but we know that the cytokine can release by immune cells. Immune cells can attack the heart muscle cells, and then they remove and replace by connective tissues. Although it is too soon to conclude the same approach would benefit men who are losing their Y chromosome, says Forsberg. Okay, here it is when they use this, when they use animals for this experiment such as mice, while it is too early to make a clear contribution, then how antibody prevent effect of transforming growth factor beta and scaling of the heart. So two questions. One is antibody, other one is why it is too early to make a conclusion. First, we address how antibody work, and it is unit 4, immunity. So you have learned antibodies can prevent heart scaling by neutralizing transforming growth factor. So antibodies specifically bind to TGF beta, and TGF beta blocked. So then antibodies specific to transforming growth factor bind to TGF beta, and no more TGF beta, and no more scaling. Too early to determine that the same method would be prevent. So it worked with mice. Preliminary investigation worked with mice, and why it is not going to work with the human. So then points are mouse research results are not always directly translate to the human due to significant biological difference, such as so metabolic pathways, immune response, and drug metabolisms are different. And the mouse genetics are not exactly same as the human genetics. DNA errors, paragraph number 9, DNA errors during cell division, which could be Y, loss of the Y chromosome is linked with the cancer. So DNA errors during cell division, which could be Y, loss of Y chromosome linked with, what is the link with the DNA error and cancer? So when the mutation happens of the genes which is responsible for cell cycling, cells try to divide uncontrolled fashion, and many cells form because many genes which regulate cell cycle mutated. Then this has linked with the cancer. So DNA errors during cell division, which could be Y, loss of Y chromosome linked with the Y cancer. So loss of Y chromosome again linked with the cancer. So model answer is DNA mutation are a significant driver of cancer development, primarily due to their impact on cellular process and the accumulation of these errors over time. When DNA errors affect gene that regulates cell growth, cell division, and repair, they can lead to uncontrolled cell proliferation. Uncontrolled cell division, repair mechanism lost, and mutation happen in genes control cell cycle. Then they continue the research in the future with XYY chromosomes. So I think we can understand why this is their next plan, because they have done so far with XY, only Y chromosome lost. Here, when one Y chromosome lost, there are 2 chromosomes remain. Or they want to check whether both chromosomes lost or one chromosome lost. So this XYY syndrome is known as a Jacob syndrome. Now, Article 2. Essence of man, Y size doesn't matter, it's Article 2 topic. So then it explains evolutionary, there is no X and Y, but when with the changes happen, the homologous chromosome, one jumped out of that as become Y, and then continuously they they underwent so much of changes to become a Y chromosome. Some offspring get landed with chromosome full of damaged gene and are eliminated by natural selection, whereas others inherit undamaged copies and survive to reproduce. I think this is again a common question. So natural selection is when mutated genes are there. So these mutated genes maybe adapt to the changes, or some are not performing well. These organisms cannot adapt, and these organisms are eliminated. By doing that, genes also eliminated. Then again, evolving of Y and X chromosome about 100 million years ago, the line of mammals that branch away from the platypus and echidna section of contained the gene variant for maleness got flipped back to front. So that is actually the one they're going to say that the Y chromosome inversion like things. So when they are evolving, one chromosomes jumped and this become the Y chromosomes and they start to make the gender. The human version now has 78 genes, far less than in original 600 or so. At this rate of decay, Y ought to disappear altogether within 5 million years, as famously predicted by a few years ago by Jenny Gravis. So, question, the human version now has 70 genes, far less than its original 600 or so. At this rate of decay, the Y ought to disappear altogether within 5 million. How this prediction made? So prediction make done by the calculation of the gene decay. So they check gene decay from the beginning, how many gene lost with the time, and they make the rate. So they make the same rate in the future to calculate the loss of completely Y chromosome. So just to explain it, by calculating the gene decay rate. So I explain it like this. 600 gene, 44 million years ago, 600 genes. Then 14 genes per million years, 1 million years, 14 gene lost. Then the rate divided by the remaining gene, 78 G, so around 5.5 million. That's how the prediction made in this scenario. Then again, 16 paragraph, further explaining about the X and Y. Next, what does the research indicate about the future of the Y chromosomes? So despite the prediction that the Y chromosome might disappear altogether within 5 million years due to its decay, there is a growing evidence that what remains of the Y chromosome is important. And so as I told you, Y chromosome prediction is eliminated in 5 years. But others say, no, there are some new genes are coming. New genes are coming due to mutations. New genes are coming due to the duplication of the genes. That means, let's say, one gene is duplicated, one chromosome is duplicated like small part, and the duplicated one further can change the genes and the sequence, then they can make a new genes. So new gene making, like things we are not learning the theory, but the prediction may be wrong because new genes will adapt to Y chromosomes. So again, they found burst of gene loss directly after inversion happened, followed by long period of stability. In fact, not a single gene has been lost from the oldest part of the human Y in the past 44 million years. Now, here is again, I'm taking the textbook, A's textbook lesson, not A2, A's textbook lesson. During crossing over, mutation can happen because when part of the DNA removed from the homologous chromosome, this doesn't happen really within the gene. I mean, if when the gene separated and part of the gene in the original chromosome and part of the chromosome in the removing college chromosome, then the sequence lost. This is called mutation, gene mutations. So gene errors. So this is in the textbook. Second, errors in the process lead mutation, and this is further way of introducing new combination into the genetic makeup of the species. Explain how gene loss take place during inversion in cell. Chromosome inversion can occur during meiosis, prophase 1, chromosome crossover, chromosome break or flips around and reattach to the opposite orientation. That means 180-degree change. This can lose genes if truncation happen within the genes. Oh, that's something, just your background knowledge, not for the articles. So new gene can arise from several mechanisms: gene duplication, transposable element protein domestications, lateral gene transfer, gene fusion, and de novo originations. This is too much, but just something like we all know, the new genes arise because of the duplication and further changes. How scientists claim that not a single gene has been lost from the oldest part of the human Y in the past 44 million years. So answer is, scientists determine gene stability in the oldest part of the Y chromosome through comparison of the human Y chromosome with those of other species. Comparison, you can use the PCR and chain electrophoresis, and we can compare the gene sequence of the other species by analyzing the gene content and structure of the Y chromosome across the different lineage. Then they can pinpoint when genes were likely lost or gained. Next, this question, a team led by the Hendrik Kaiserman at the University of Lausanne, Switzerland, surveyed the Y chromosome of 15 different mammal species and one bird. Now, question is, why this study include one bird with 15 different mammals? The answer is to compare the effect of gene loss of other animals apart from mammals, to find the maleness evolution of other mammals, what is the relationship of how they happen in the other animals. Then the regulatory gene retained in the Y had to be so precisely calibrated that organism couldn't survive the outward intermediate stage. When this work around did not yet function perfectly, such as James Turner at the RMC National Institute of Medical Research in London. Regulatory genes are particularly vital because they cannot control many other genes. How regulatory genes control other genes? Question, answer. Regulatory genes control other genes by producing transcription factors. Transcription factors bind to promoter, then RNA polymerase bind. Transcription factors can either activate or repress transcription of the target gene, thus influence the amount of mRNA and ultimately the amount of protein produced by those genes. The last 20 which happen in about 80% of elderly men. Now, if everything now we discuss happen in a people, losing the Y in some tissues take a toll. Chromosome can be lost when cell divide, and men who lost Y chromosome in their bone marrow. So bone marrow also the Y chromosome lost. This happen in 8%. Then have a higher risk of cancer and die in average of 5.5 years younger than other men. This can lead cancer and they can die early. Why this happen only in 8%? It's a question. Answer is other factors. Example, environmental factors and other genetic factors. So this is contribute to become this 8% again, like epigenetics like things can also play a role. So not all 8% develop this because other factors can accelerate the other people, some other factors which prevent it. So that's end of the discussion of the scientific article, and go through again this PDF file and the questions, and good luck with your exams.